Welcome, everyone, to this week's edition of the Extracellular Vesicle Club. This is actually our 172nd edition, and we're very privileged to learn today about how we can discriminate between EVs and some other extracellular particles, specifically lipoprotein particles. I'm actually at a meeting in Korea this week, and we just heard today from a group that was talking about, actually Edit Buzesh's group, who found that lipoproteins can associate with EVs in some situations. And so it's very important for us to be able to understand what is a lipoprotein particle and what is an extracellular vesicle. So we have Deun van Deun and Vahid Sandoghdar today, who are going to be presenting some very interesting work that they have done. And Vahid, I think you're going to begin. Is that correct?
That's right.
All right. Well, please feel free to share your screen, and you can begin whenever you're ready. Thanks so much.
Okay. Yeah, thanks, Ken. This is great. Welcome, everyone, to our little presentation. So I'll be speaking the first few minutes, and we'll tell you about the method, interferometric nanoparticle tracking analysis, in short INTA, and Deun will then continue to tell you actually about the interesting stuff that was just alluded to regarding the lipoproteins and EV separations and distinctions. All right, so I believe nanoparticle tracking analysis, or NTA, is quite well known in the community of EV scientists. This is a commercial method, and it's based on dark field microscopy, or any way that you can get intensity from particles. So you can do this, for example, in formal fluorescence shown up here, or dark field as shown here. So imagine that you have a suspension of any type of nanoparticle, and if you were to illuminate that in a way that you only look at the light that is scattered by the particles, then you would expect to see these particles jiggle and wiggle, and this is indeed what you would see. So this is the kind of a video that you could record from particles that are undergoing Brownian motion in suspension. And now, because you look at individual particles, you can track them, chase them, and as you see here, for example, the individual trajectories can be recorded. And once you have a trajectory, you can find a mean square displacement of these particles and look at that as a function of time or delay, and you get a curve that you can fit. And that way you can, following this simple equation, you can figure out what kind of diffusion constant you have. And the diffusion constant is directly proportional to the slope of this curve. Now, one knows from statistical physics that the diffusion constant is proportional, indirectly or inversely proportional to the size of the nanoparticle. And of course, it also depends on the viscosity and temperature of your suspension. But by doing this, people have been extracting the size of particles for quite a number of years now. Again, also in commercial device, and has been applied many times. So this is the kind of signal that you might be getting out of this. That is, you look at a large number of particles, and each time you figure out the size, and then you can plot histograms and learn about the size distribution in your sample. Now, the intensity that you record is proportional to the 6th power of the particle size. I'm not going through the details of why this is, but this is what you want to remember. And this signal also depends on the index of refraction of the particle and the index of refraction of the medium. But in the formula that I just showed you, one only worries about this size d. But because this index of refraction information is there as well, some groups have actually tried to get more out of the data and can say something also about not just the size distribution, but also about the scattering cross-section, that is the overall brightness of the particle, and not just how fast it moves around. And by looking at the brightness, you can also say something about the different indices of refraction that your particles might have in a suspension. For example, Van der Poel and colleagues could tell the difference between silica and polystyrene particles. And similarly, you could, for example, look at EV in blood and not just look at the size distribution, but also say something about the index distribution. So this method basically works. And again, it is used quite commonly by many labs. So it's easy and fast to use, and it measures the hydrodynamic size. So the size information comes from the motion of the particle. And if you are careful and look at the exact brightness of the particle and calibrate that, you can also get information about the index of refraction. But the problem with this method is that because the size of the signal depends on the sixth power of the size of the particle, there is a very big difference between smaller particles and larger particles. So the whole method is usually skewed towards larger particles because the signal from larger particles dominate. And just to get a sense of the sixth power, if you were to compare the signal from two particles that are only about twice different in size, the actual size is about 60 sometimes different. And of course, if you have a larger size signal, you can track that particle with better precision, and therefore you get the diffusion constant and the size of the particle better than if you have a weaker signal. So that translates to having not very good trajectories, and not having very good trajectories also means that you can't go very fast. You don't have a lot of signal to look at details of the motion. Now, in the method that we are using, INTA, we're doing basically the same thing, except that the signal that we get comes from interference. And interference is familiar to everyone who might not even be a physicist. So the electric field of light, has a wave behavior, so has this cosine function. And as a result of that, you can have two waves interfere destructively or constructively. On the left, they would do a constructive interference. On the right, they would do destructive interference. So again, that's high school physics that you're being reminded of. And the way interferometric scattering microscopy works, as opposed to dark field microscopy, is that you now, again, illuminate the sample, but instead of dodging the illumination, you actually collect light that is coming back from the sample. Let me get a laser pointer. That is coming back from the sample, and the light that comes back from the sample interferes with the light that is coming from the particles. So you have particles in the sample, and you have this substrate here, and they both give light, and the two interfere. So the light that is reflected from the sample is kind of like a reference. That will give us one intensity, and if you just look at the cover glass, you will get an image like that. And now if you put 20 nanometer gold particles on the cover glass, the overall signal is going to look like this, because it's the sum of the electric field from the particle and the electric field from the substrate. The two add up. And you can then write the same thing for the intensities. It's not so important right now. But again, the point is that the contribution that is only coming from the particle scales as the 6th power, and at some point becomes negligible for very small particles compared to this other term, which is proportional to the electric field that's scattered and not the square of the electric field that's scattered. And therefore it scales as a 3rd power of the particle size. And that means that now the difference between a certain particle and something that is twice larger is only a factor of 8 and not 64. So we're not going to be discriminating against the smaller particles as quickly as we were in dark field microscopy. So the way the actual experiment works is that you know your background, for example, from the substrate, and if you now subtract these two, you get beautiful signals from individual particles. It is a very strong signal, even if you have a small nanoparticle, and that allows us to do nanoparticle tracking analysis with much better precision and higher quality. Okay, so here's now an example of the type of snapshot of a video that you might record. In one field of view, you have different particles. Some of them are out of focus, some of them are closer, but we can track, we can localize each one of them, figure out their centers, and then basically track them in time, establish trajectories as you would do in NTA as well, and do the same type of analysis, determine the size of the particle, and make histograms. And when you do that, then you see that in, for example, for a model sample that has 30 nanometer gold particles, NTA would give you this broad distribution. INTA would give you a much sharper, tighter distribution, which is much more comparable to the results of electron microscopy, scanning electron microscopy, or transmission electron microscopy. So we've benchmarked this in this paper that was published last year in Nature Methods, where we presented the method the first time. We've gone through a few different case studies. One of them is a mixture of 30 nanometer, 20 nanometer, and 15 nanometer gold particles. And so these are, first of all particles that are usually smaller than what NTA would be comfortable with detecting. But on top of that, now you have a mixture, so you want to distinguish them. And in INTA, we can do this very nicely. One thing that we do regularly in INTA is that we do scatter plots in one dimension. That's just the hydrodynamic diameter or radius that we extract from diffusion. And on the other axis, we plot the third root of the contrast. And the third root of the contrast has the same dimension as the size of the particle. And but these are two independent measurements, and therefore gives us more information than before. And you can clearly see on this plot that we are recognizing 3 different populations. If you were to only look at the measured diameter, then you would not see the 3 populations. You would think that maybe there are 2 populations, but in the contrast, we see 3 of them, and also in the overall representation. Okay, so something like this is recorded only in a few minutes, but it's very easy to make these measurements also for much longer times. Right, and here now is, if you want to compare this, what you would get from the same sample, but using standard NTA. And you see here that the 3 populations are not at all resolved. Another case study, we've mixed polystyrene particles of 2 different sizes. Again, we can recognize them as 2 separate populations, not just because we've colored them, but because you can clearly recognize these different populations, clouds, if you want. Here's an example where polystyrene particles and silica particles and gold nanoparticles were mixed of different sizes. And again, you can clearly distinguish them. A kind of a muscle flexing version is shown here, where we take 10 nanometer particles, but then 10 nanometer particles that have been pegylated, and 20 nanometer particles and 20 nanometers that have been pegylated. And you can see that we comfortably identify all of them. So again, the signal that we have now is it has information about the size as before, and the index of refraction. It looks just like the signal that you had for NTA, except that the NTA was square of this, and this is what we have for INTA. So by making these measurements, we can identify subpopulations of very small nanoparticles. And I now hand over to Jan, who's going to tell you about the EV measurements.
So thanks, Vahid, for this introduction to INTA. So indeed, when Vahid came to us, which I guess is about 2 years, maybe a little bit over 2 years ago, with this technology that they developed, so this really cool technology. I mean, the first thing, of course, that I thought about as a EV nerd, basically, is how can we use this to, improve the detection of EVs, and especially EVs from plasma, because plasma is a really difficult case for EV detection, as I've tried to depict here in this sort of artistic impression. So detecting EVs in plasma is basically a really difficult game of Where's Waldo. So you have, of course, some of these Waldos wandering around in the plasma, but most of it is, of course, free protein, but also a lot of other particle types. So lipoproteins mostly, which outnumber EVs, as you can see here. So it's really this soup of lipid particles, and maybe also some protein aggregates where the EVs float around in, but it's of course really difficult to look at the EVs themselves. And then even after isolation, usually the image looks like this. So there's still a lot of lipoproteins there, which might interfere with the detection of the actual EVs. So the question that I had in my mind, knowing that INTA can, have these independent measurements of both the size and the refractive index of particles, is the INTA able to distinguish between the EVs and the lipoproteins? So why do EVs and lipoproteins have different light scattering properties? Well, as you probably know, so EVs have a lipid bilayer and then a lumen inside, with some proteins, RNAs. But all in all, it's like a pretty sparse particle, let's say. While lipoproteins, these are these really densely packed packages of proteins and lipids, which are known to scatter more light than the EVs, because they don't have this lumen inside. So this is then basically the first experiment that we did together, where I had some EVs purified from melanoma cells, so melanoma cell culture. I purified them by density gradient, just to be sure that we are, have a really pure population of EVs. And this is then what we get with the INTA measurement, so the diameter on the x-axis and the contrast on the y-axis. And then we did the same measurement basically with commercially bought chylomicrons and VLDLs. So these are the two lipoprotein populations in the plasma which overlap in size with EVs. So these are often quite difficult to remove using size-based isolation methods, which are often used in plasma, like SEC, so size exclusion chromatography, or even with ultracentrifugation, because they will show similar pelleting behavior as EVs because of their overlapping size. So as you can see here, indeed the size is overlapping with the EVs, but then the contrast looks different. So you can see that the clouds, already look different. And then when we match this to the refractive index that we would expect for these particles, we see that this is quite different. So for the EVs, the refractive index is usually below 1.4, which has been published by other groups as well. While for these lipoproteins, because they are more dense, they scatter more light, this is usually above 1.4. So this was the first indication that it's probably possible to discriminate them using this INTA technology. But of course, these are you can transform this then from contrast to refractive index. So basically showing the same thing, that the size is overlapping, but then the refractive index shows a completely different pattern between EVs and then these lipoprotein particles. So when we measure the same particles with NTA versus INTA, we see basically what Vahid talked about earlier. So with the NTA, you usually have a bias towards larger sizes of particles. So you can see that all of the size histograms are sort of skewed towards larger sizes for the NTA, or for the INTA, you also pick up on the smaller particles. Okay, so this was then for the individual populations, but then we also wanted to see how well does the INTA perform when we have mixtures of particles. So we have either just the EVs, or only VLDLs in this case, and then we also have the mixtures. And then you can nicely see when you have less and less EVs, more and more VLDLs, that you have this this population here. So with the refractive index above 1.4, which pops up gradually. And indeed, as expected, with increased proportion of LDLs or VLDLs, we get more of this population. Then we measure the same mixtures using normal NTA, and then plot the diameter versus the intensity. Yeah, you get these clouds. So it's a bit similar as also what Vahid showed with these artificial particles or synthetic particles, that you can really distinguish between these two populations with normal NTA or conventional NTA, let's say. So using these measurements, we can also build a classifier. So I think this was a random forest classifier, and then we chose a cutoff of 80%. So we only sort of track the particles where we are at least 80% sure they are either VLDLs or EVs. And then we can quantify this, so and plot the expected EV concentration, so based on the dilutions we made, and then also versus the measured EV concentration. And we could see that this correlates really well, and we have like around a 4% error rate, at least when we have no EVs. Then we still measure 4% of the lipoproteins as being EVs. So this is something that you can't really avoid, but I think this is an acceptable error margin as opposed to conventional NTA, where the error margin is basically 100%. What you can also do with INTA is then look at the individual diameter and refractive index of these two populations. So the EVs are generally a bit larger in this case than the proteins, and the refractive index also, in these different mixtures stays the same. So it's able to really distinguish these populations and extract a refractive index, which is below 1.4 for the EVs and then above for the lipoproteins. Okay, so this was just EVs and VLDLs, but of course there's also these other particles. So we also made mixtures of EVs with then chylomicrons, VLDLs, and LDLs. So again, we see a very similar pattern, where we have these clouds of LDLs, of lipoproteins that appear when you have increasing proportion of lipoproteins. Also, again, with the NTA, you just see clouds, which you can't really distinguish the different particle types. Again, the classifier that we built, and then here you see the result, where we again have like a 3 to 5% error margin of the expected EV concentration versus the measured one. And in this case, there was a bit more overlap in the diameter between the lipoproteins and the EVs, because we also have these chylomicrons, which generally are a little bit larger than just the VLDLs. All right, so this was then for the sort of artificial mixtures, but we also wanted to test it in real world samples. And there we made EV preparations using size exclusion chromatography, but also with another chromatography-based method, which is capable of extracting or removing a bit more of these lipoproteins, which is dual mode chromatography. So this is published a few years ago. So with dual mode chromatography, you don't have just the size exclusion layer in your column, but also a cation exchange layer. So the point is that these lipoproteins, they have this ApoB protein, which has a lot of positive charges. So these can interact with negatively charged beads, and then these particles will be delayed, so as they flow through the column. So this dual mode chromatography is then a way to not only remove the smaller particles with the size exclusion part, but also then the ApoB containing lipoproteins with this cation exchange layer. And in the end, you will get more pure populations as with SEC only. So this is shown here. So visually, you can really see when you start off with, let's say, a quite lipemic plasma sample, that in the SEC you have a lot of these lipids, while in the DMC then it's a lot less. Also in electron microscopy, so all these white particles are lipids basically, so they're removed with the DMC, more at least than with the SEC. When you do the ApoB ELISA, you can see that DMC is able to deplete more than 99% of these ApoB containing lipoproteins. And then when you load these on Western blot, also the EV markers, they pop up a little bit more when you load equal particles. Because of course, this is equal particles by conventional NTA. So the conventional NTA also measures the lipoproteins as EVs, or as particles. So when you load equal particles, you basically don't see your EVs as if you only use a SEC, because most of the particles you measure are actually lipoproteins and not EVs. All right, so what we had was a plasma sample from a volunteer before having a fatty meal, and then 3 hours after a fatty meal. So where we would expect many more lipoprotein or much more lipoprotein contamination after the fatty meal. And indeed, this is what we found. So on top, the INTA measurements of the DMC isolated samples, and then at the bottom, the SEC isolated samples. So you can see mostly these blue particles, which are then classified as lipoproteins, and only a small proportion of these red particles, which are supposed to be classified as EVs. Indeed, we did an ELISA for ApoB. So you can see pre and postprandial, indeed, there's an increase in ApoB concentration, because of course, after the fatty meal, you will have more circulating lipoproteins. And also there's a really, really big difference between the SEC and the DMC. So this is, the DMC is able to remove about 50-fold more ApoB containing lipoproteins than the SEC. So one point here was, this was a healthy volunteer, which didn't appear to contain so many EVs, or this sample didn't appear to contain so many EVs. So as you see here at the bottom right, so the relative EV concentration was below 4%. So there was only below 4% of particles really classified as EVs, which is actually our error rate, which we determined in these artificial mixtures. So we're below the error rate, so these are probably not really EVs that we are counting there. So just to show that the INTA is still able to extract the EVs or identify the EVs in these samples, we spike them again with these cell culture EVs. So in this case, 5 × 10⁻11 particles per ml. And now you see this nice, red population of EVs popping up in all the size refractive index plots. Of course, a little bit less with, or a lot less, I would say, with the SEC isolated samples, because of course, they contain the most, especially postprandial, they contain really a lot of these lipoproteins. But if you can see here on the top right, the dashed lines here indicate the expected EV concentration based on the measurements without the spike. And you can see for most of them, especially both DMC samples and also the SEC sample before the fatty meal, you can see that this corresponds quite well, so that the INTA is indeed able to distinguish this spiked EV population from the lipoproteins which are already present in the sample. Okay, and then finally, to have also a sample with endogenous EVs present, we had a melanoma plasma sample. And this was also postprandial, so this was a really highly lipemic sample. As you can see here, I took a picture of the EV fraction after SEC. So this was basically white, so there was just so many lipids in there. And this is also shown in the INTA graph, so where you find just almost only a population of lipoproteins and this really low population of EVs, which is again below the classification error, so it's only a 3.7% EV population there. And then using known recovery rates or published recovery rates, we could go back to the original EV concentration in the plasma. But then we also did the same for DMC, where we had a higher relative concentration of EVs. So again, there we calculated back to the concentration in plasma, which is, as you can see, a lot lower than with the SEC, estimated by SEC. And then as positive control, or let's say like the gold standard technique for EV isolation or really purification of EVs, we also did a density gradient isolation. And then you can see that you have this really nice, population of almost only EVs with very little lipoprotein contamination. And also there, when we calculated back, we came to plasma concentration originally of about 6 times 10 to the 10th per milliliter. And this agreed indeed with the one we found with the DMC, while the SEC is really overestimating the number of EVs, because we are at this misclassification or under this misclassification rate. So this is not really a reliable measurement. But at least when with the DMC, the lipoproteins were outnumbering the EVs by up to 1 order of magnitude. So even then we can still accurately quantify the EV population, as we show them with compared to the density gradient control. All right, so just to summarize, the INTA is just as easy and fast to use basically as the NTA. Right now we get the results, or at least Vahid's team developed it so that we get the results in real time. It's also similar to NTA. It can measure the hydrodynamic size, but also then the refractive index and the concentration of particles. Vahid's team is also now working on trying to get it to measure fluorescence, which is already working quite well. It has a higher sensitivity than NTA because it's proportional to the third power of the diameter and not the 6th power, as with NTA. Dynamic range is also high. It has a more precise size determination as conventional NTA. Some downsides is that it creates large volumes of raw data, but I guess this is just basically a hardware issue. It needs a fairly concentrated sample, so we measure in these small nanoliter size drops. So of course, to get a proper measurement, you need a proper amount of particles or traced to get statistically valid measurement. The accuracy is still dependent on the EV to LP ratio. So this is what we showed actually with these SEC samples, right? So we have a lot of lipoproteins. So if this outnumbers the EVs by more than 20-fold, basically, then you still have this misclassification because there's just a little bit of overlap of the refractive index between the lipoproteins and the EVs. So this is something that at least at this point we can really avoid. And then it doesn't exist as a commercial instrument, or at least not yet. Some things we are working on. So as I already mentioned, Vahid's team is working on fluorescence or adding fluorescence to these measurements. So we really want to combine this with labeled, so labeling the EVs for different markers and combining the INTA measurement with that. We have a lot of clinical samples which we want to measure, so to really accurately characterize them, both in terms of size and concentration, which was not possible until now, or at least not with the conventional NTA, because you always are measuring particles, but you don't really know if they are EVs. At least with the INTA, you have a much bigger certainty that you are really looking at the EVs themselves and not on other particles. Some improvements that we thought of is that we could introduce maybe measurement under constant flow, so you have a more representative image of the whole sample. Single particles had a potential determination to further characterize the particles, or the EVs could also be of interest. You can always increase the laser power to get an increased sensitivity, and go maybe more in depth into the data analysis to further find how we can lower the, let's say, misclassification rate of EVs or of lipoproteins. Okay, so with this, I would like to thank the first authors of the paper, so of Vahid's group. So this is Anna and Martin. So Anna also made a lot of the slides you saw today. So special thanks to her. Then Marie and Jan Ole. So Marie from Vahid's group as well. She did a lot of work. Jan Ole from our group, and then Andreas, my PI. I'd like to thank our funding agencies, and then of course also you. For joining us today, and if you have any questions, then we'll do our best to answer them. I think Ken had to leave, so I will sort of take over for the discussion. Maybe Ikyun, if you could unmute yourself, and then you can ask your question.
Hello, can you hear me?
Yes, we can hear you.
Thank you for very nice work. Actually, I have 3 questions. Is that first one is, can you observe the movement of particle in the real time before you guys doing the subtraction of background?
Yes, to a large extent you can, but these are of course very fast. So, but you can definitely see the particles, especially if they're larger. I mean, at some point your particle becomes very small, and there's no limit to how small it can get. But you typically see the particles in real time.
Okay. The second question is for the experimental setup, do we need to use the very narrow line width laser which has very narrow line width for the coherence or to see the interferometric pattern well?
Yeah, no, you don't have to have a very narrow band laser. You measure usually a few microns away from the substrate, and the only thing you care about is that the coherence length of the laser would be large enough to be of the order of a few microns. So that's not a very narrow band laser.
Okay.
Because then the light that is scattered is going to be coherent enough with the light that is reflected from the substrate. And so you don't have to, almost any laser that you have do the job.
All right. So you are saying the coherence length, that mean you are talking about the collimated beam?
No. If you think in terms of the bandwidth of the laser,
okay,
then the bandwidth you can convert to a time, coherence time.
Okay, all right,
and the coherence time gives you the coherence length.
All right.
That is over what length the photons have the same phase. That's what I mean by coherence.
Okay. Oh, okay. Thank you so much. And the final question is about, actually, I was trying to make some build some setup for iSCAD. So I feel like it was pretty hard for me to make focus the beam to the backside of objective lens and to get very well collimated beam, because following the reciprocity is pretty hard. Yeah. So I don't know if it's possible or not.
Yes, your question is a good question, because according to textbook kind of optics, you would expect to go in the back focal plane and be perfectly collimated. And typically you can't get it to be perfectly collimated. But if you see that your beam doesn't diverge by a lot over something like a meter or so, that's good enough. Because the light that comes back doesn't have to be perfectly collimated. It just has to be collimated enough to keep its intensity to give you a signal.
Okay.
If you have any questions, feel free to write an email and we can walk you through the details of getting your setup to work.
Wow. Okay. Thank you so much. Yeah.
All right. Thank you for your question. Then I guess Sasha is the next one. If you can ask your question.
Hi, thanks. That was really, really interesting. So if I understand it correctly, like this would be very useful for optimizing EV preparations. That's correct. So regardless of the method you use, we could use this technology to know what proportion of our sample actually has EV versus lipoprotein. Is that right? In a nutshell?
Yeah, exactly. So
Yeah, great.
Yes,
So
but I just add that, indeed, this is like a really, fast method, so you don't have to do, or necessarily have to do like the Western blots for the EV markers, which, can be kind of tedious. So you can really have a really quick look at what your sample looks like, how many EVs you have, how much contamination you have. And
I
then,
think
yeah.
it's really, really helpful. I do have a couple of quick questions. Are you, I think it would be great for, in general, people to know the relative proportion of EVs that are in a given prep. So are you, or have you, I'm sorry, I'm not familiar with your literature. Have you gone through some of the prototypical presumed EV isolation or enrichment methods and from various biofluids, and then sort of document what proportion of those samples actually have EVs versus lipoproteins? If you haven't, I think that would be very useful for the field. And my second question was, you demonstrated a Western blot where I think you showed with your dual phase setup, you removed ApoB, but is it correct ApoA1 is still present in the dual phase? And have you looked at just HDL by itself? Because I noticed you didn't have any HDL or LDL specific profiles. Where are they on that spectrum of lipoprotein like scattering or profile?
Yeah, so yeah, your first question. So we did not systematically analyze like every EV isolation method at this point. So the ones that are included in the paper are then, SEC, which is, I guess, the most used by many people in the field right now. Then the DMC, and of course, then the density gradient sort of as a gold standard. So I think those three already cover a lot of the used methods. Ultracentrifugation is something we haven't tried, so I try to avoid centrifuging for several reasons. I mean, it's hard to get it reproducible, really. It's a lot of work. You lose a lot of EVs. Your EVs can, deform and everything. But this is definitely something, we could think about doing in the future, just to show, how pure the samples are when we do the ultracentrifugation method on plasma. Then the second question, so we did also look at, well, we didn't actually look at HDLs, but we looked at LDLs, which are already a lot smaller than VLDLs. And there we saw that we actually do not detect them, at least with the current setup. So we could adjust the setup to also look at these smaller particles, but in this case, I mean, EVs, they don't go below 30 nanometers, which is sort of the size of LDLs, and HDLs are like about 10 nanometers, so even smaller than this. So we weren't really interested in measuring these particles because we are interested in measuring EVs. So we didn't change the setup to allow measuring these particles. Mm-hmm.
That's also very, actually, from my personal experience with collaborators, I think that's also very important to communicate, if you have not already, that they fall outside of the sort of your dynamic range. And then the major, presumably, or sorry, they're falling outside of the dynamic range of the NTA. Is that what you mean? Or the INTA?
Yeah, basically, yes.
So
but they also need.
Yeah. Sorry, go ahead.
I mean, they're also typically less of an issue because they are, a lot smaller, so they don't really get pelleted so easily. Also, the SEC, I mean, it's aimed to remove smaller particles, right? So depending on the type of resin you choose, this is typically like everything below 70 nanometers or 35 nanometers should be removed, which includes especially the HDLs. So that is why, I mean, it's known to remove most of the HDLs already. So that's why we didn't really focus on this population. I think the VLDLs are much more, and the chylomicrons are much more of a contaminating population than the HDLs with the currently used methods, at least.
Thank you very much. That was a very good talk. Thank you.
All right, thank you. Then we have Elena, who has a question. Maybe she's not there anymore. So I can also just read the question. So in order to measure the samples obtained in SEC by INTA, did you have to concentrate them further? If yes, how? So yeah, indeed, when you obtain these EV fractions after a SEC or after any chromatography-based method, we usually do an ultrafiltration, like a 10 kilodalton cutoff ultrafiltration to concentrate the sample and then get it, have it measured with the NTA or INTA. Then Steve has a question.
Hey, just wondering, I mean, this is something you guys put together yourself, right? This is not a commercially available instrument yet?
That is correct. We are now making non-profit prototype versions that we deliver to different groups so that they can get going. There might be a commercial version of it in a year or two on the horizon, but right now, if you're interested in having an instrument in your lab with which you can measure, please feel free to contact us, and then we can possibly put something together for you.
Awesome. Very, very interesting, very promising. Thank you for your work.
Thank you.
Okay, so then we can move to Christina.
Hi there. Very nice talk. Thank you. I did a little bit, much more rudimentary work. I actually did precipitation, and then I had a friend, Kim Bruce, who was working on HPLC isolation of lipoproteins. And we kind of concluded in a very, I guess, almost deductive reasoning that they were together. I think it's really important, though, the work that you're doing, because we need to think about whether when we're doing cohort studies, do we need to have people fasting? Because it seems like the conclusions, if there's always lipoproteins present. And so do you think there is always lipoproteins present? Is there ever a situation, or any of the techniques I guess you've used so far, they always seem to be contaminating, in inverted commas? And what can we conclude from our data thus far if that's the case?
Yeah, so I think from what we showed, I think only the density gradient is really able to remove at least the majority of lipoproteins. But of course, this is quite a tedious operation. So a density gradient has to run overnight, and you have to collect the fractions, and yeah, it's not really possible to do this in any type of clinical or preclinical study. So this is the only method that we found that was able to remove them up to a point where you can measure your sample also with let's say regular NTA, because most of the particles you measure will be vesicles. I think with any other method, which is also what a lot of people have shown until now, with any other method, especially size-based methods, you will always have the lipoproteins just because they overlap in size. So there's no way to remove them. If you only select for size, you will have them there. So in that sense, fasting samples will definitely help.
Super helpful.
Yeah.
Thank you.
This was okay.
No, it was a beautiful talk, and thank you for your answer.
Thank you. All right, Rachel, if you want to ask your question.
Hi, thank you for such a really nice talk. Yeah, I was basically just wondering if it's possible to do these experiments if you have a commercial NTA. Do you know if this is compatible with any kind of commercial setups or no?
I think commercial NTAs wouldn't be suitable, but if you have a commercial microscope, it wouldn't be too difficult to convert that into this type of device. Particularly NTA is not, the NTA devices are just because of the way the illumination works and all of that. The optics is set up is not, I don't think that it would be a good idea to adapt that. But again, if you have almost on any optical platform, you could imagine doing this. So if you are interested, again, feel free to get in touch with us and we can walk you through it. And part of the thing that is there is not just the optics. The optics is actually not very complicated. It's the data analysis, and would be happy to provide you with the codes and all of that.
All right, thank you, Vahid. Then I think the final question is by Martin.
Hello.
Yes, hi.
Hello, everybody. So, what factors affect the refraction index of EVs? The components, the media, also some pre-analytic variables like time it's been in the fridge or Darwin fridge in cycles.
Yeah, that's a good question. I think this is something we don't really have the answer yet, because we didn't really investigate this at this point. So I can only say we isolated EVs from different cell lines, for example, and then we always found like a very similar pattern, at least in terms of size and refractive index. Or at least, you know, when you look at the median size, median refractive index, this was also always very similar. I would expect, you know, if you see some smaller variations in refractive index, I guess this has something to do with their contents. So whether they contain maybe a bit more protein, maybe a bit more nucleic acids. But yeah, this is, you know, this is something we could potentially investigate by creating sort of like designer vesicles, or, you know, vesicles, synthetic vesicles with some more lipids, some less lipids. I don't know, or some more protein or RNA. I don't know, Vahid, if this is something you have already done, or
yeah. So I wanted to say a couple of words about that. So from a biological point of view, you know, lots of things could be contributing to the index of refraction, but from a physics point of view, the index of refraction is something that if you read, I recommend that you look a closer look at the paper that we published, because the index of refraction is a kind of a lump quantity that we would be talking about. Actually, when you look at EVs, you have to worry about the index of refraction of the interior and the index of refraction of the lipid shell that would then would contain proteins or not, and then the thickness of the shell comes in. So there's actually a lot more to the overall construct than just an index of refraction. So when we plot just one index of refraction, that's in a way a simplification, and we have a more of a detailed discussion of this in that paper. Of course, because there are lots of parameters that go into the signal, it's not very easy to decipher all of them independently, but that would be something that you might want to look at if you're interested in pursuing the question of index of refraction.
All right. Thank you, Vahid. Thank you, Martin, for the question. I think these were all the questions. So yeah, just would like to thank everyone for joining us today and for your interesting questions. And if you have any more input questions, then please reach out to us via email. I think we're always happy to help. So thank you, and see you next time. Bye.
Thank you.